Colorectal cancer is expected to be third most diagnosed cancer in Canada in 2022. It is expected that 10% of all new cancer cases will be for colorectal cancer, and 11% of all new cancer deaths would be as a result of colorectal cancer. So assume that this is a DNA of a cancer cell, and after getting treated by either chemotherapy or radiation therapy, the DNA will be damaged. Later on, an ERCC1-XPF as a heterodimeric enzyme complex with endonuclease activity will participate in the repair of DNA double-strand break that was produced as a result of either chemotherapeutics, such as platinum-based drugs, or ionizing radiation. And then a cancer cell resistance happens. So inhibitors of ERCC1-XPF dimerization, such as small molecules, can disrupt this interaction between ERCC1 and XPF, which will lead to sensitization of colorectal cancer to the effect of genotoxic therapeutics. And then the DNA, the damaged DNA will remain damaged, and then as a result, the cell would die. So we have made an exciting discovery that a drug that is clinically used as anti-malaria, pyronaridine, can inhibit the interaction between ERCC1 and XPF. You can see the structure of the drug on the right side of the slide. And on the left side, you can see a test that we did to actually investigate or confirm the fact that actually pyronaridine has this effect. The test is PLA, proximity ligation assay. And on the top row of the picture, you can see the control group. And on the bottom row, you can see the group that was actually treated with pyronaridine. You can see the nuclei of the cells that are stained with DAPI and are shown as blue dots, and the interaction between ERCC1 and XPF as red dots. And in the merged picture, you can see that this dimerization is actually happening inside the nuclei of the cells. So after getting treated with pyronaridine, you can see that there are barely any red dots appearing, which means that the dimerization between ERCC1 and XPF is inhibited. So our hypothesis is that liposomal formulation of pyronaridine can act as novel inhibitor of ERCC1-XPF heterodimerization, sensitizing colorectal cancer cells while sparing normal tissue to the effect of platinum-based chemotherapeutics. Our objectives would be first to develop an optimized liposomal formulation for passive tumor targeting, and second of all, to examine the anti-cancer activity of free and encapsulated drug in colorectal cancer models in combination with platinum-based drugs. So our liposomal delivery system was made out of 3 phospholipids, DSPC, DSPE-PEG, and cholesterol, and the preparation method was thin lipid formation. The drug was loaded by remote loading using a pH gradient. So outside, the liposomes hold a pH of 7.4, and inside there is the acidic pH of 3.5. The unloaded drug is separated from the loaded one by ultracentrifugation at 150,000 G for 2.5 hours at 4 degrees Celsius. The size of the liposomes were assessed by dynamic light scattering using Zetasizer. Encapsulation efficiency and drug content was assessed by disruption of liposomes by 4% SDS using UV spectroscopy at 426 nanometer. And in vitro drug release was assessed by putting the samples inside dialysis bag and immersing dialysis bag into release media, which was made out of PBS and 4% BSA. Samples were taken from inside the bag, and the test at different time points, and the test duration was 72 hours. The cytotoxicity of the loaded and free drugs were assessed in HCT116 colorectal cancer, human colorectal cancer cell line. And the chemotherapeutic agent were oxaliplatin and carboplatin, and the possibility of synergistic effect was assessed using cost-benefit formats. So you can see that the size of the formulations, both the drug-loaded and free liposomes, are around 100 nanometer. PDI is something around 0.1 or 0.2, which is totally acceptable. Loading content is around 15%, and the encapsulation efficiency is more than 90%. As for the release study, you can see that the blue graph is demonstrating the release of free drug. Most of the drug is released in free version around first 2-3 hours, while the formulation holds the drug inside for more than around 72 hours. Around 50% of drug has been released. As for the cell studies, cells were given the Q4, the cells were treated with Q4 in 8 different concentrations. And then 4 hours later, they received 12 different concentrations of carboplatin. Same concept was done for oxaliplatin as well. You can see here the darker the color of the matrix, the lower the cell viability. And here is only the dose response curves based on the two that are obtained based on the 2 treatments. And later, the cost-benefit format using Lowy model were able to assess the possibility of synergism in both loaded and the free form of pyronaridine while being treated with carboplatin and oxaliplatin. As it is obvious, you can see a synergism specifically when the cells are treated with carboplatin with loaded form of pyronaridine, when they're treated with 0.15 micromolar of carboplatin and 3.25 micromolar of carboplatin. So before wrapping up, I want to thank my professor, Dr. Afsan Al-Awsanifar, Dr. Michael Weinfeld, my previous lab mate, Dr. Sam Sadat, and all my lab mates, NMIN, Alberta Cancer Foundation, and University of Alberta. And in conclusion, the lipid-based nanoparticles loading for pyronaridine has showed an average diameter of 82 nanometer, which is suitable for passive tumor targeting. Liposomal formulation has showed excellent loading of pyronaridine and showed that the release in slowed the release in biologically relevant media, and also it demonstrated that there is a potential for pyronaridine and its liposomal formulation in chemosensitization of colorectal cancer to platinum-based chemotherapeutics.
Okay, then may I ask you something, Parnia?
Sure.
Will you please explain again the mechanism of the drug loading?
Yeah, sure. So the drug has different the pyronaridine has different ionization forms, the groups. And as you can see, like when the pH is acidic, it has one carboxyl group and 3 different amine group that gets protonated. And because of this effect, it will get more lipophilic. So when the pH is acidic, the drug has a tendency to pass when it passes through the lipid bilayer because it has a higher lipophilicity. But when it gets through the core of the micelle and gets encounters the acidic pHs, the lipophilicity of the drug decreases. And because it decreases, it does not have the tendency to get out again. So most of the drug gets trapped in, while the outside pH is 7.4, close to neutral. So because it has lost its lipophilicity, it doesn't get leaked out. We use the lipophilicity, the tendency of drug to load the drug inside the liposomes.
Okay, yeah, thank you. That's great. And well, I could ask another question meanwhile. What will be your future steps?
So since we saw synergism in the mentioned concentrations, we're going to do colony forming assay to assess that, because cytotoxicity assay is not enough to confirm the DNA of a cell has been damaged, because the cell could be still alive during the 72 hours, but has lost its ability to proliferate. So what we do is we will perform colony forming assay to assess the activity of the drug in a longer period. And if we see the same results, then we'll head to animal studies to actually see if the loaded version is showing a better effect compared to the free version.